We start off with with pathology. We touched a fair bit on this morning, and in terms of, and I think this is a very exciting area of brain tumour management and recognition of different types of tumours and different ways they should be treated. And Michael Buckland, who's a clinical associate professor at the University of Sydney and works at Royal Prince Alfred, and he's also been instrumental in setting up Brainstorm, one of the charities that's been so generous to us today and help this event occur. Michael's going to speak about molecular tests and pathology and the process of pathology and how that's all done. So if you can join me in welcoming Michael.
Thanks, Jonathan. So, yes, I'm going to talk about basically tests related to the diagnosis of brain tumour. And as a pathologist, I deal with tissue. So we're not talking about interpreting MRI scans here. We're talking about once you have an operation, whether that be a tumour resection or a stereotactic biopsy, the piece of tissue that comes out is sent to the path lab. And I'm basically just going to take you through what happens in the path lab to arrive at a diagnosis. So what I do in my job, there's a couple of different roles. First of all, in some operations, when the surgeons aren't entirely sure what they're dealing with, they might stop the operation halfway through and send a small piece of tissue down to us to have a look at immediately so we can tell them what's going on. Now that often a situation might be, we're not sure if this is a high-grade glioma or a lymphoma. Let's take a little bit out and work it out. So we don't necessarily give them the final diagnosis then, but we can guide them in the right direction. This is a high-grade glial neoplasm versus, oh no, this is a lymphoma, you shouldn't cut it out, they need radiotherapy. And then the next thing I do is, of course, once the operation's finished, the rest of the tissue that's been taken out is fixed in formalin. Generally, it's fixed overnight, and what fixing does is it cross-links proteins, but the idea of fixing is that the tissue is then stable, it's not going to rot, so then we can play with it. And on that fixed tissue, everything that's cut out is sent to us, and then we provide a final diagnosis. And then, based on what we see down the microscope, we then often will recommend or do some of these ancillary molecular tests that I'll talk about as well. And then, of course, an important role is communicating with the surgeons, the oncologists, the radiation therapists, often in multidisciplinary meetings. So as I've said, the intraoperative consultations, they send us down a small piece of tissue. Often the questions are, is this lymphoma or glioma? Is this a high-grade glioma or is it a metastatic cancer? Or is this actually a tumour at all? And sometimes patients, when they present with multiple sclerosis, they can have something called tumour-factive demyelination, which looks almost identical on the scan to a high-grade glioma. So that's something we always have in the back of our mind. Is this actually a tumour at all, or is it actually demyelination, first onset of multiple sclerosis? And this is some pictures of what happens in the lab, and actually this isn't my lab. I've borrowed these from another neuropathologist from Brisbane. Two things we do. One is we get a very small piece of tissue about the size of a match head and squash it between two microscope slides and then slide it out, and that's called the brain smear. And then by sort of dipping it through a whole variety of different jars like this, we stain it up, and then we can look at it under the microscope like that. And you get a beautiful look at the individual cell morphology that way. And then the other thing we do is we freeze a tiny little bit of tissue in liquid nitrogen. That makes it hard enough that we can cut very thin sections from the tissue. And we're talking 5 micron thick sections. They need to be so thin because our microscopes, the light's underneath, we put the tissue there, and then we look at it up here. So it has to be thin enough for the light to go through it. And that's called an intraoperative frozen section. That's not too dissimilar to the final pathological assessment. We do something very similar once the tissue has been fixed in formalin. It's important, just as a side note, in terms of the intraoperative consultations, they're a preliminary report. So if you've had an operation and the surgical team the next day tells you the diagnosis, or the pathologist says it's that, that's actually the preliminary report. The actual formal report takes a week or two. So, and up to 10% of the frozen section diagnoses will change in the final report. So generally the final diagnosis you get is when you go back to the surgeon a few weeks later, then they will have the formal pathology report. Often the diagnoses that are changed, it's not a complete change of diagnosis, it might be a change in grade or something like that. But it's important to remember that the final diagnosis does take a week or two. The formal pathology is based on this book, the WHO classification, and it's all based on what the tissue looks under the microscope. And it's actually been that way since the 1920s. And obviously it's not exactly the same as it was in the 1920s, but it's still based on this is what it looks like under the microscope with a couple of routine stains. Now, it is all about to change, and I think it was alluded to this morning. The WHO is convening committees at the moment to work out how best to incorporate some of these new molecular markers into the formal classification of tumours. And it's been recognised that just because a tumour looks one way under the microscope, it doesn't necessarily mean it's all tumours that look like that are exactly the same. There might be a few different types of tumour that to us down the microscope look very similar. So it's a work in progress. We have had some publications that guide us in what we think the WHO is going to do, but we're expecting that in a year or two, the new classification. Nevertheless, most modern pathology labs in Australia are already incorporating this sort of stuff into their pathology report. And I'm going to talk a little bit about that in a minute. So the tumour names, some of them are fantastic names, and it's always fun saying them because you sound intelligent. They're named after their resemblance to normal brain cells. So the glial cells in the brain, so in the brain there's neurons, also called ganglion cells, and they're the ones that do all the information processing. But then they are surrounded by all these supporting cells. These cells that basically nurture the neuron, make sure it's in place. If there's a damage to the brain, these cells can produce their own type of scar tissue, and they're the glial cells. And there's different types of them. And the most common one is the astrocyte. And if the tumour down the microscope looks like it's composed of astrocytes, we call it an astrocytoma. And, or if it's a very high-grade tumour, then a glioblastoma. The oligodendrocytes produce myelin, which is the fatty coating over the nerve processes. And if it looks like oligodendrocytes, then we call it an oligodendroglioma. And then there's also ependymal cells, ependymoma. And then some more uncommon tumours are called ganglion cell tumours, gangliocytoma, ganglioglioma. And that's because part of the tumour actually looks like neurons. So that's where all the names come from. I've just got a couple of examples here. As you can see on the left, an astrocytoma. This is what it looks like under the microscope. And one of the most important things is that we see these fine processes just like normal astrocytes in the brain. And on this side, and it's a little bit maybe overexposed, but I hope you can see that if you look at individual cells, so those round blue circles, they do look a bit different. And on the right there, that's an oligodendroglioma because they look like normal oligodendrocytes in the brain. So once you have the tumour type, okay, this is an astrocytoma, you then have to give it a grade. And a grade is a rough indication on how that tumour is going to behave. And the WHO has a very simple grading system that's applied across lots of different tumour types, not just brain tumours, but sarcomas and other tumour types. And it's a 4-tiered grading system. In the brain, grade 1 tumours are thought to be discrete, and they're thought that if you can cut them out, they won't come back. If you can't get them all out, they might come back because you haven't been able to get them all out. But if they do come back, they generally come back as the same thing. So particularly like maybe a meningioma, a grade 1 meningioma. If you can't cut it all out, it might regrow. But if you cut it out again, it looks the same. It's a grade 1 meningioma. And so that's a grade 1 tumour. If you can cut it all out, it's potentially curable. And then there's grade 2, 3 and 4. And in the brain, these really refer to the common gliomas. The common gliomas aren't so discrete. They tend to have cells infiltrating off the main tumour mass. So in fact, it's impossible to cut it all out. And then based on a variety of features we see down the microscope, if we can see cells dividing, if we can see cells dying, if we can see vessels proliferating, these sort of histological characteristics all then feed into the grade and then we will call it a grade 2, a grade 3 or a grade 4. And in brain tumours, a grade 4 in gliomas is the glioblastoma. So it's important to know you've got to get the tumour type right before you can apply the grading criteria. So something like tumour cell death in an astrocytic tumour tells you it's a glioblastoma, a WHO grade 4. Tumour cell death in a meningioma, a WHO grade 1 meningioma, means it's a WHO grade 1 meningioma. It doesn't necessarily change the grade at all. So you can only interpret those grading features once you know what the tumour type is. So we have tumour type and grade, and that's what forms the basis of any pathology report. So I'm just going to move now into some of the molecular markers that are sort of used fairly routinely now in clinical practice. There's the IDH mutations, the ATRX mutations, 1p19q codeletion, and MGMT promoter methylation. Now, these days, IDH mutation status and ATRX mutation status can actually be assessed at the time of that formal pathology report. So generally, in the week or so after your operation, when you have the formal pathology report, the IDH and the ATRX status should be on that report. These other tests take longer because we have to do extra fiddly things, and so often they will come out a week or two after the formal report with those ancillary molecular tests. So IDH stands for isocitrate dehydrogenase, and they're enzymes, and they're actually involved in energy metabolism. And it's certainly a growing theme not only in brain tumour biology, but lots of different tumours. People are starting to recognise that in fact metabolism and metabolic pathways, which generally have not been thought of to be critical for tumour growth, are actually very important in tumorigenesis. So there's this intersect of metabolism with neoplasia, which is certainly an interesting area of research. So IDH mutations, they're very common in the grade 2 and the grade 3 astrocytomas and oligodendrogliomas. And we use them for two reasons. The main reason is it actually helps us make a diagnosis now. Sometimes in tumours that look a bit ambiguous down the microscope, if we're able to detect an IDH mutation, that'll push us, we know now this is definitely a grade 2 astrocytoma, it's not something else. So it's incredibly useful diagnostically, and it also seems to have some prognostic value in that the uncommon tumours that don't have an IDH mutation do tend to behave a little bit worse. But that data is somewhat preliminary, and people are still arguing about the exact prognostic implication of not having an IDH mutation. From my point of view as a diagnostician, I use it to help nail the diagnosis. And we're very lucky in that these mutations, they don't stop the protein being produced from the gene, but it results in the production of an abnormal protein. And then we can stain for that abnormal protein on our microscope slides, which is why it's easily incorporated into a standard pathology workflow. We're used to putting stains onto microscope slides, and this is just another one we can do. And this is on the left here, that's the picture I showed you before of an astrocytoma. And on the right here, this is an astrocytoma stained for the abnormal IDH protein, and the stain comes up positive. So then we can say, oh look, we don't have to do any more, we don't have to even do complicated DNA sequencing. That stain tells us that this tumour is harbouring the mutation. Now the stain works for about 85% of cases, but there are about 15% of cases where there's a slightly different mutation and that stain does not work. So in those cases, we do then have to go down the DNA sequencing pathway, in which case that IDH mutation status might take a few extra weeks after the formal pathology report. But generally that's in the report saying we're doing sequencing and we'll issue another report. So the next thing is ATRX mutations. Again, incredibly useful from a diagnostic point of view. In the past, the only way we could tell something was an astrocytoma versus an oligodendroglioma was how it looked down the microscope. And now we have a few different markers that actually assist us by saying, oh, actually this is showing astrocytic differentiation. So ATRX mutations are found almost exclusively in astrocytic neoplasms, not oligodendrogliomas, typically not the typical glioblastomas either, but the grade 2 and 3 astrocytomas. So this helps us confirm the diagnosis of an astrocytoma. And these mutations, rather than producing an abnormal protein that we can stain for, they actually stop the protein being made. And the ATRX protein is expressed in every cell of the body. So so what we then do is we look for loss of expression. And we do another antibody stain. So again, just another stain on the microscope slide. And it's completely different now. So this is overexposed, but I'd point out to you that these very faint blue dots, these are actually the tumour cells. And they're not taking up that brown stain, which means they don't express ATRX. So with IDH, we're looking for the presence of a stain, means there's an IDH mutation. With ATRX, we're looking for absence of a stain. You can see there are some brown dots in the background, but not many. And that's the normal brain cells that have been overtaken by the tumour, and they still stain up because they're not part of the tumour, they're the normal brain. So those two stains have really revolutionised the diagnosis, an accurate diagnosis of brain tumours. This 1p19q loss in oligodendrogliomas has been known for quite some time. Rather than IDH and ATRX, which have all just sort of come out in the last 5 years, it's probably been 15 years that we've known about 1p19q codeletions in oligodendrogliomas. And what it means is that actually the tumours lose the short arm of chromosome 1 and the long arm of chromosome 19. So it's not a specific gene change, it's actually a chromosomal change. There's millions of genes not millions, there's thousands of genes on the chromosome. And we're not talking about any one gene, we're talking about that whole chromosomal arm. And it's very common in the oligodendrogliomas, the grade 2 and 3s, about 70 to 80%. So again, we can use this in a diagnostic sense. Well, we think it's an oligo, let's look at the 1p19q. Not as useful as the others, because remember, only 70 to 80% of oligodendrogliomas have that genetic lesion. So if you get a positive result, oh yeah, this is 1p19q co-deleted, that's very helpful, because then it's confirmed it's an oligodendroglioma. If you get a negative result, so it's not 1p19q deleted, well, it's not so helpful, because we know that probably 20 or 30% of oligos don't have that marker. But still, it is certainly useful in helping confirm the diagnosis. And there is some prognostic value as well. Certainly, it's thought that the tumours that have 1p19q loss tend to grow slower, and they tend to respond better to chemotherapy. And so, really, I think our practice is that anything that even smells faintly of oligo, we'll do the 1p19q just to make sure, because we don't want to miss one of them. Look, I know there are oncologists in the room, this is not my area, but in terms of changing practice, well, in a low-grade oligo with 1p19q, the oncologist might decide to hold off treatment. Say, well, it's got the good prognostic markers, let's just sit back and watch. It might not need treatment for another 5 or 10 years. Or if there's no 1p19q codeletion, they might decide, okay, we need to hit it now. So it does feed into that sort of treatment algorithm in many instances. And this again is still a stain on microscope slides. So we cut the tumour very thin, put it on microscope slides. It's another different stain, but this one is a fluorescent stain. So you have to look at it under a fluorescent microscope in the dark. And we are looking at green and red dots. So each of these circles or ovals is a tumour cell nucleus. And then we look at say the red dots and the green dots. And the green dots is actually chromosome 1Q. And the red dots, so that's the other arm of the chromosome, and the red dots is the chromosome 1P. Now we know everyone has two sets of chromosomes because you get one from your mum and one from your dad. That's why you're 50% of each of your parents. So everyone has two chromosome 1s. They have their dad's one and they have their mum's one. So normally if we do a probe for a chromosome, you'd expect to see two dots. That's normal. But when we lose the short arm, the 1P, the short arm of, say, chromosome 1, we only get one dot. So this is a probe for chromosome 1. The red dots is 1P, the green dots is 1Q, the other arm which doesn't get lost. And it's telling us that, oh yeah, there's two, in these cells, there's two copies, as per normal, of 1Q, the other arm, but there's only one copy of 1P. So that's how we'd say, oh yeah, there's 1P lost there. I hope that makes sense. All right, and then the final thing I'm going to talk about is MGMT promoter methylation. MGMT is a DNA repair enzyme. It's very important in normal cells to help keep our DNA intact and to repair any damage from ionising radiation or funny chemicals. It's a very important enzyme. But in a tumour, when we actually want to try and damage the DNA with chemotherapy or with radiation, we're trying to get the tumour to die, and one way of doing that is damaging the DNA. In the tumour, the presence of MGMT is actually not whatever damage you do, it then goes and tries to repair it. So if MGMT is switched off in the tumour, as it is sometimes, then it's thought that the tumours respond a bit better to chemo, particularly to chemotherapy. And MGMT promoter methylation is a way of measuring whether or not that MGMT has been switched off or not. It's particularly relevant in the glioblastomas, much more so than the other lower grade tumours. And in glioblastomas, those ones with MGMT promoter methylation tend to respond better to chemotherapy. Now, in most instances, I'm stepping again out of my zone of expertise, but in most instances, someone with a glioblastoma will get chemotherapy and radiation therapy. But in some patients, I'll come to that in a second, but in some patients, it's fairly harsh treatment, as you probably know. They can't handle both the chemotherapy and the radiation therapy, and so the oncologist decides, we're going to step back from that standard protocol. It's too harsh for this patient. Let's take a modified protocol. And in those cases, knowing the MGMT methylation status might guide the oncologist's choice in chemotherapy versus radiation therapy. This test has been dogged by laboratory problems. It's a finicky test, and getting reliable and reproducible results is quite difficult. So it's still there's still a bit of controversy in the literature, but it's something that some people want and some people ask for, and we do it and other labs do it as well. And we measure ours through a machine called the pyrosequencer, and we get this sort of readout. So again, this is moving completely away from the microscope into a DNA sequencing machine, but certainly an important test. Luckily, we're able to, because when we fix the tissue with formalin, it actually, we can still extract DNA from it. So by putting the tissue into formalin, you're still able to get DNA out, so you can do these other DNA-based assays. It doesn't have to be all down the microscope. And as I've said, it's really only relevant in glioblastomas, and it might guide the choice of radiation versus chemotherapy. Okay, so that's all actually I was going to talk about today. We do have a website where I think you can, most of these tests are there, and if you need to contact us, all our details are there as well. Thank you.
What we might do is just hold off on questions for Michael at the moment. We've got sort of a bit of time after Kerry's talk for questions for both of them. So we'll move on to hear from Kerry. About 10 years ago, I came out of doing my neurosurgery training and spent some time in the lab doing research, and Kerry had just come from working on rock melons and started learning about brain tumours. I'm sure there's some laboured joke about melons there, but I'm not going to go there. But so it was it's the start of what's been a very good relationship over the last 10 years or so, and Kerry's gone on to become one of the most, if not the most accomplished brain cancer researchers in this country, and I'm very pleased to have her talk today about research development and early phase clinical trials.
Try and keep that rock melon stuff a secret, Jonathan. But if anyone wants me to pick out a rock melon, I know just the trick to do that. So it's always good to have other talents, isn't it? So what I'm going to talk to you today about is research development and predominantly early phase clinical trials. And this isn't particularly just for glioblastoma, it's for all tumour types that we can work with. So what is the problem with treating a brain tumour? Well, the biggest problem is its diffuseness. So as you would have heard from Jacob this morning, it's very difficult to resect a tumour that big and that diffuse. And on the right-hand side is what I call the little fella. It's one glioma cell that's driving through the matrix. Where did the 2-year-old go? He would have loved it. Something for him. Well, it's good for all of us. So yeah, it's just one single brain tumour cell. So you can see why they're very difficult to treat, is because they're so infiltrative. The other thing which Michael just spoke about is DNA repair proteins. So a lot of the chemotherapies we use actually target the DNA in cancer cells. So what they do is they nick the DNA or nuke the DNA. And so then the cells can't repair themselves and then they die. Cancer cells have this mechanism, which Michael was calling MGMT. They have this mechanism where they can actually repair the DNA and then continue growing. So that's a big problem that we need to overcome, is these repair proteins that the cancer has. And then one of the biggest factors, and I think this is something to really glean on hope, is the fact that every patient is different. There's not one patient that's the same, there's not one patient that behaves the same, and certainly the tumour, and Michael can attest to this, looking down a microscope, there is no tumour that's the same. So when they give you a diagnosis of a median survival of 15 months, tell them to stick it, because not everyone is going to be 15 months. You're going to have long-term survivors there. The other problem that we've got, though, is intratumoral heterogeneity. So we're all different, our tumours are different, but inside the tumour we have different variable bits of the tumour. Some bits are glowing green, some bits are glowing red. So it's very hard to treat with just one chemical. So knowing this intratumoral heterogeneity, that means that we have to start combining treatments. So that's why radiotherapy and temozolomide work as a combination, not one after the other. And you'll find that a lot of the new drugs on the market will be in combination with something else. And this is a really good way forward. Okay, so I'm going to talk about a subject that's not mine, but it's one of the hottest topics for patients at the moment, and that's immunotherapy. So I first apologise for the images that I'm going to put up, because I drew them. And again, the 2-year-old would have been better at doing this than what I am. So here goes. Immunotherapy, it's not an easy subject, and certainly it's not one entity. Okay, so here's my dodgy slide. Okay, so that's a tumour cell on the right, and that's an activated T cell on the left, just in case you're not sure. And the aim of immunotherapy is to get that T cell to attack the tumour cell and kill it. So that's essentially what immunotherapy is. So immunotherapy can come in all different packages. So the first one is that it could be a peptide or a tumour cell vaccine. And so these are vaccinations that you get off the shelf. One of the good examples of a vaccination is the EGFR variant 3 vaccine, which is sometimes called Celdex, or the CDX 110. What it does is you inject it in, it stimulates the dendritic cells to then activate the T cells, and then the T cells will start eating the tumour cells. So there's been a big phase 3 trial that's just run at the moment, and we're waiting for the results. Another example of immunotherapy is actually an Australian invention, and that's using monoclonal antibodies. And what we need to find is something on the tumour cell that's not on the normal cells. And if we can find something that's unique to the tumour, then we can make monoclonal antibodies, which then hone in on that tumour, target it, and then again the T cell can come in and eat it and get rid of it. This is a little bit more complicated, which is the cellular immunotherapy, and it's usually a treatment that's used for blood-borne diseases like leukemia. Essentially what we need is part of the tumour or blood to be taken out of the body, and then what we do is make T cells and dendritic cells, make a vaccine in a chest tube, and then put that tube back into the body. So it's a little bit of an invasive process. The other thing we can do is what we call CAR-Ts, or CARs, and that's basically genetic engineering of T cells and then putting them back into your body. I'd have to say that this therapy hasn't been developed well for brain tumours yet, but there is potential. Now, one of the most promising areas of immunotherapy now are these badly drawn checkpoint inhibitors. So, what the tumour cell has, which is so annoying, is that they have these programmed death ligand receptors, and they bind to the T cell PD1. And this binding actually suppresses the immune system, and the cancer cell has done it deliberately so that it can evade the immune system. But we're better than the cancer, remember, and what we've come up with is fantastic checkpoint inhibitors that can actually block this interaction between the tumour cell and the T cell, and therefore allow normal killing of the tumour. So I think this is one of the most amazing inventions in our time. And only in the last 12 months have we seen huge breakthroughs with melanoma, lung cancer, breast cancer. It's such an exciting field. And I think one of the good things to know is that brain tumours have a lot of this PD receptor. So theoretically, it should work. Okay, so I just wanted to quickly mention the clinical trials that have been going on at the moment. So we've got the rindapepimut, which is the CX110 or Celdex, and at the moment there's a Act4 trial going on, which is a phase 3 trial. They've stopped recruiting and we're just waiting for the results. But one of the interesting things is that there's been 3 trials to precede Act4. They're really ingenious with their names, Act 2, Act 3 and Activate. And they've got one in recurrent gliomas called React. But you can see that the median survival with temozolomide and radiotherapy was 15 months. You can see quite quickly here that they've already increased the median survival by at least 5 months. And I think the really exciting thing is the percentage of survivors over 3 years, which is over 25%, which is fantastic. So this is really promising treatment coming out of Duke. One of the other things that they've just published from Duke University is most of us, 95% of us, harbour a cytomegalovirus or CMV. And so our tumours also have this virus. And so what Duke University has done is made a vaccine against the CMV virus. But what they found was if you just give the patients a shot of tetanus or diphtheria, Why not? The dendritic cells actually migrate to the T cell a lot quicker, and the results have been phenomenal. Absolutely phenomenal, just by adding an extra vaccine in there. So that was published in Nature about a month ago. Now this is an Australian invention, which is ABT414. And this is the monoclonal antibodies recognising EGFR variant 3. Now don't get too worried with this slide. I sort of looked at it for a few hours and then I thought, who knows, who cares what's going on here. But the core of ABT414 is that it actually recognises only tumour cells have an upright receptor. Normal cells don't. So this antibody recognises that. Now this is what we call a waterfall plot, and this is pretty exciting plot, and this is only phase 1 data. And what you can see from the waterfall plot is that it's missing its x-axis, its numbers, all that sort of stuff, but you don't need to worry about that. What you need to worry about is, or get excited about, is these lines going down. They're tumours that have shrunk. So we're looking at a tumour that's shrunk by 100%, tumour that's shrunk by 70%, 50%, 60%, so on. So this is really exciting, and as I said again, it's an Australian invention where we're seeing complete response in 1 patient, partial responses in 3 patients, and stable disease in another 3 patients. So this is really cool, and this is going to be coming out in clinical trial really soon. And then I mentioned about the checkpoint inhibitors, ipilimumab. Now I wish these people, these pharmaceutical companies, would stop calling these drugs ridiculous names. But an antibody always ends up in a mAb. So we've got ipilimumab, which has done wonders in melanoma. And now I want you to look out for these 2 new drugs, which is nivolumab and pembrolizumab. Remember those because they'll probably come your way quite soon. And these 2 are PD-1 checkpoint inhibitors. So it's quite exciting. Okay, chemotherapy. Yep, so chemotherapy has had a long history, and now we're moving into more targeted therapies. So this is a pathway which I'm not going to go into, thank goodness. But you can see that there's multiple drugs targeting multiple parts of the pathway. And we're not just targeting genes, we're also targeting metabolomics. And so one of the things we know is that your cancer cells actually breathe differently to normal cells. And we can tap into that, and we've got some drugs that can actually change the metabolism of a cancer. And DNA repair, as I mentioned, it's one of those things that with strong DNA repair in the cancer, a lot of our drugs don't work. So a good theory is to target that DNA repair mechanism. And one of those mechanisms is called PARP. And PARP repairs the single-strand breaks when we give them chemotherapy. So what we're doing is a clinical trial, and that's going to start in June this year. And this is actually going to take patients that are non-methylated, so unmethylated GBM patients, and they're going to be treated with a PARP inhibitor called veliparib. Now that PARP inhibitor is then going to be combined with radiation. And so my lab has actually come up with some really good data showing that the radiation and the PARP inhibitor work beautifully to kill the cancer cells. And then the patient's going to receive temozolomide after that. And then we'll follow them up. So as I said, this trial is actually going to start in June, which is good. Where my lab comes into it, we've come up with a signature that we think patients with this signature will respond best to this treatment. Now, maybe some of you have heard the term sequencing or whole genome sequencing. Now, whole genome sequencing is basically where we take the tumour and we sequence absolutely everything in that tumour. So we have a map. We have the map, the design of what that tumour's made of and what's gone wrong in that tumour. And this is what the map looks like. And these are circus plots. And around the outside of the circle are your chromosomes, and then these little lines that streak in are all mutations. And as you can see, G54 and G89 are completely mutated to the buggery. So you would have thought that this is a bad thing, but it's not. Because they've got so many mutations in them, we hypothesised that maybe if we used a DNA repair drug, we could knock them over. And that's what we've been able to do, is actually take these tumours, treat them with the PARP inhibitor, and completely abolish the tumour. So now we're going to move forward and use this signature in the clinical trial that I just showed you, but they're also going to be using them in the clinical trials run in the UK as well with PARP inhibitors. So that's cool. Okay, now I just want to finish up on my favourite subject, which I believe is going to be the future. And I harp on it a lot about how we are all different. The tumours are all different. And we shouldn't be receiving the same drug as our next door neighbour. Every patient should be tailored for their treatment, and that's called personalised medicine. Okay, so what we do at our lab, which is over at the Lowy at UNSW, we work with the surgeons at Prince of Wales Private and Prince of Wales Public, and Jacob's one of those surgeons that we work with. And when a patient comes in, we get a call and we go up to surgery and we have a chat and wait a while and then wait some more. and then when the surgeon's ready, we take the tumour and we bring it back to the lab. We also take a blood sample or later on a saliva sample because it's absolutely critical that we get your normal DNA as well. So we're looking at tumour DNA and normal DNA. So some of it goes into the freezer up the top there, and that's what we call Tumor Banking, where we put it into the bank and then you can access it at any time during your diagnosis. But we also take a piece of that tumour and we dissociate it, which means just break it up into little pieces. And we put some of those little pieces into a culture dish and grow your tumour cells. And some of those tumour pieces actually go into what we call a surrogate. So you've all heard of surrogacy and women carrying your baby for you. Well, this is mouse surrogacy, where the mouse is carrying your tumour for you. Now, that's a big point that we're trying to make here, is that we've got your tumour in a dish and we've got your tumour in a surrogate. Because then what we're going to do, or what we do, oh, that's just a science thing, don't worry about that, is we look at the overall survival and we do this whole genome mapping. And the whole genome will tell us what's overexpressed or what's underexpressed or what's the problem, and then we can treat accordingly. And we mentioned this morning about repurposed drugs, and that's where the repurposing of drugs comes into it, is that you may actually show an aberration that's been treatable in multiple sclerosis. So having your tumour in the dish and having the tumour in the surrogate, we can then go and test it for you without putting you through a clinical trial. And then if it works, well, you're the first person to know about it, and we'll tell your clinician. And so these are the little babies, they're little mice, and that's the tumour that we typically see. And so I just wanted to show you a real life example of how this is working for patients. Now, this patient came in with a bad headache in 2013, and she had an MRI and it was completely clear, which was odd. She had really bad headaches. The symptoms started to progress and she started to not be able to read words on a recipe. So she had another MRI in 2014. This is the tumour that developed in a period of just less than 6 months. So as you can see, it's a pretty hideous GBM. What we did was we took the tumour, we went through the whole process, and then we started to treat the tumour. And this is the most exciting stuff to come out of our lab, is the fact that we've been able to kill this tumour. And we've been able to report back to the family and the clinicians that we may have drugs that will help this patient. So on the left, the patient's really responsive to the PARP inhibitor, and on the right, she's also responsive to a combination that we've come up with temsirolimus and PNAO. So this is really cool stuff. Okay, so I'd just like to finish up by reminding everyone of a epidemiology study that I spoke about this morning, and that's AGOG, which is a family case control study of glioma. And it is what it is. We're investigating the possible causes of glioma, so whether it's lifestyle, genetic, environmental or occupational. And we're looking at a whole range of targeted exposures, and we're also listening to you as well. You may have your own theories, and we're certainly open to those theories as well. And it's a paper-based questionnaire, and we make no apologies for the fact that it's huge. And that's because we don't know. It's an exploration study. We're trying to find the cause. So I'm glad it's big. We ask for a blood sample, and we're consenting to approach a sibling or a spousal partner. So the aim of the AGOG study is to around about 800 patients and 800 normals, and we're about halfway there. So on that, I'd just like to finish by acknowledging my lab that are all sitting over in the corner, so if you want to annoy them, go ahead. All right, thank you.
Take a seat, Kerry, and Michael, do you want to come back up too? Well, thanks, Kerry. That was very interesting and very educational. It's nice to know where the sort of cutting edge of this stuff is going. Do we have any questions for Michael or Kerry? Over there, Jacob. It's interesting to see that we're obviously, Kerry's taking the personalised medicine thing to the nth degree, which is great, and it's obviously something that I think will come into more widespread clinical practice. But I think a lot of what Michael was talking about with the more simple, more widely available stains is actually very, very important and very useful, and there's a degree of personalisation within that as well. So I just think that should be noted. Sorry. Can you just the switch on the side?
Michael, I'm just wondering with the slides that you hold post for biopsies, I think you actually did mine too, coincidentally, are they able to be reused down the track? So if you then go on to do, is it sequencing and things like that, can you just use the one
Sure.
they took out last July, or do they need to find some more?
That's a very good question. We archive the tissue and there are national pathology accreditation guidelines that say every tissue sample that comes out of a patient has to be kept for a minimum of 10 years. The tissue that comes out, for the formal pathology, we actually embed it in paraffin wax, and that's how we can get those very thin sections. So generally, if it's a reasonable resection, we have only shaved off maybe only 10 or 20% of the tumour, and the rest of it's still sitting in that paraffin wax. And that paraffin wax block goes into our archives for a minimum of 10 years. At the moment, we're keeping them for 30 years, and that's available for future testing. It all really depends on how much. If you've had a resection, then we hold a fair amount of tissue. If you've had a small stereotactic biopsy, then most of it might have gone in the diagnosis. But regardless, we keep whatever's left, and we'll keep that for you. And so the idea is, if there are new tests that come up or new treatments, or you want to take part in a clinical trial, we can go and retrieve that specimen and do what we need to do.
And Kerry, I assume they can't then be put into rats once they've been in paraffin.
I knew that was a double-edged question, wasn't it? So with paraffin tissue, what we can, there's limitations to paraffin and what we can and can't do, but certainly the block is an amazing archive of DNA, and that DNA can then be used for the sequencing. So we can certainly do the sequencing side of things, but to put it back in the mouse, unfortunately you'd have to go back into surgery, which we wouldn't ask you to do. So, but we can certainly do sequencing on that tumour.
Could I just ask one question about the personalised medicine? So, and establishing the DNA for the individual tumour and all that sort of thing, is that available for peds as well, or is that only available for adults?
We certainly don't. Our personalised medicine is in conjunction with the biobanks that we run. So in the private, we have consent to do paediatrics, and in the public, we've got consent just to do adults over 18. So it just depends on the consent type that we've got.
And would that be for your trials as well?
Yeah, that's right.
Yep. Okay.
Yeah.
Thank you. Yeah, it's just a question for both of you, really. If you had a tumour that was just diagnosed or in the process of being diagnosed right now, would you, or perhaps you had a child who had a tumour, would you insist on molecular profiling for that tumour, regardless of whether it was being offered or not? And if you then had it done, for instance, and you found out that the tumour was MGMT negative, would you proceed with, say it was a glioma, would you proceed with temozolomide or would you not?
That's a really good question. And I have to say I can't give an unbiased answer to that because of what I do and what I've seen with the power of genomics. So I would be insisting that my tumour gets sequenced. I would be insisting that I find a model somewhere around the world, if not in Australia. And if the luck of the draw doesn't come back my way, so MGMT non-methylated, supposedly that means that you're not sensitive to the temozolomide. We don't know that for sure, but there's a good chance that that's the case. Barry, to be honest, I would be pushing for an alternative treatment. That would be, it's my honest opinion that, you know, I'd be pushing as hard as I can, and if my clinicians wouldn't offer me that, then I'd find somewhere else that would.
We might phrase that question to Helen later as well, to Helen Wheeler as an oncologist. Obviously that's a situation she faces most days as well, but I think it's a good one too because there are other strategies for getting around the resistance that MGMT offers as well, so perhaps Helen might cover that in her talk.
Could I just add one more thing? I mean, that's one thing that I guess didn't point out in my talk. The new WHO classification, finally there's a realisation that in fact paediatric brain tumours are completely different beasts to adult brain tumours. So that's certainly an area that's evolving rapidly, and I know there has been a few recent publications about certain chromatin remodelling pathways that are abnormal in paediatric brain tumours which aren't abnormal in adult brain tumours. There's been a paper on perhaps if you again get drugs to target this other pathway, they may offer some promise. But all I've seen has been experiments in Petri dishes. I haven't seen any data for, in patients.
Waiting for the surgeons again. I don't know.
I keep Kerry waiting because I like her company.
Thanks, Jacob. No, that's a really interesting point because I know Barry tragically lost a daughter to a brain tumour, and I have a son who has had, I don't know what he is, but he had an astrocytoma. Now, I'm just wondering, are there significant differences between a paediatric and an adult tumour? I mean, why aren't the two related? I don't understand. Not that I understand much of what you've said anyway, but I'd really... Does that make sense?
I don't have a simple answer, but certainly it's not unusual. It's not just in brain tumours. I mean, we know in many cancers that the children get different forms of cancer than adults, and it might just be that their because they're growing, they have different progenitor cells, they have different stem cells, and so when they go wrong, they give rise to a different tumour type. That's theoretical, but certainly it's not just restricted to brain tumours, and it's not a clear-cut distinction either. So occasionally we'll see in 20-year-olds a paediatric signature, and occasionally in children you'll get the adult sort of signature based on our imperfect understanding of the molecular lesions as we know it. So it's not a simple divide, and it's not a question easy to answer, but it's certainly not restricted just to brain tumours. Certainly leukemias particularly, it's there are different beasts in children compared to adults.
I think what Michael's also drawing attention to is what we said this morning about the fact that these tumours can look identical under the microscope, but the harder you look at them, the more different they are in terms of the genomics and the actual genetic alterations that have led to that tumour. They may end up looking the same, but they're very, very different. And that's true within adult tumours, and that's true, there are some tumours that occur in both paediatrics and adults, and they behave differently. I think it's the more detail we can find about the individual things that lead to the tumour developing is what's going to make the biggest difference in outcome as time goes on. What we might do is, we've got a couple more questions. That's fine.
I'm just interested in the blood. You mentioned before there's a couple of times when you take blood samples, but is there currently, or is there any potential that you can determine markers in the blood in relation to brain cancer as you do, as we can in other cancers early on in the diagnostic stage?
I think I should hold the microphone here because I'd say no and
yes.
You say yes. I am a little bit hesitant to say yes because of the fact that we have so much trouble getting drugs into the brain. What makes us think that there's going to be markers that come out of the brain? Now, you've got a different opinion on this, and I still wait to be proven wrong. But markers that come out of the brain and are so dilute in the body, I still haven't seen any good evidence.
What about at the chromosome level?
I think theoretically it's possible. I think it's harder than most of the other cancers that have much easier access to the blood. And certainly we know with the other cancers, the blood-based markers are still problematic. So, there's been a huge debate about PSA testing for prostate cancer and how useful that is. So I think the brain tumours would be even harder, and I think so learning the lessons from other cancers, it's certainly not going to be simple. There has been some experimental evidence saying that in certain tumours, when you know what the genetic marker is, sometimes you can pick it up in the peripheral blood, but then translating that into a reliable blood test it's a long way to go.
Were there any more questions? We've got time for one or two more.
A question for Kerry regarding the example you gave for the personalized medicine. From the moment that you got the tumor after the surgery until the time when you developed the treatment, how long was that? And the second question is, in that example, was the patient also given radiotherapy or just that treatment alone?
That's a really good question, and an important one as well is our timelines of how fast we can do this. So the actual models and setting those up took around about 3 months, and as we get more experienced, the quicker it's going to get. And so we're doing well on that side. As far as the sequencing and actually deciding on treatments and getting it into the in vitro and in vivo systems, that took about another 3 to 6 months. So all up, this case was 12 months. that we did.
And was the patient given something to be?
Yeah, so the patient was given upfront chemo radiation therapy. And so ideally what we'd be looking at is if the patient unfortunately does recur, that's when we'll be asking for the treatment to be used.
There was one more question up the back and then we might move on.
My question is to Michael. You said that they have to keep the slides for 10 years. Is that for a private hospital and private pathologists as well?
Yes,
It
yes.
is.
It's a national pathology accreditation guidelines, a standard. So yes.
Thank you.
And the one more down the front here, sorry. Did you want to ask a question?
I was just wondering, when you're looking at the brain tissue sample under the microscope, is it always clearly defined what sort of tumour it is? Could it ever be a mixture of a tumour, or is there a time when you just can't define what sort of tumour it is? And if so, do you send that tumour overseas somewhere or to a different lab that has more specific equipment?
Yeah, that's a very good question, and it's certainly one that pathologists think about a lot. Certainly there are cases that are easy to diagnose, and there's certainly to diagnose, and there's some cases that are more ambiguous, and they can be more challenging to diagnose. We do, in very unusual cases, we do send slides to other experts in the field to get an opinion. I guess once you get to that point, then what you're asking them for is an opinion rather than a diagnosis, because there's no agreed upon hierarchy of who's the best pathologist in the world. So, and if you get an opinion that you don't agree with, you might send it around and try and get 3 or 4 different opinions, and then make a decision based on that. Most pathologists would try and convey that in the report, whether the diagnosis was difficult. I think that's an important part of the report, or the communication back to the treating clinicians, that this is a very difficult diagnosis to make, in which case they might then themselves request it to be sent elsewhere. So it does happen. We tend to send stuff around Australia. So there'd be maybe 6 neuropathologists in Australia that I would send stuff to for a second opinion. So, I mean, it happens. In terms of the molecular markers, I think there has been a great controversy in brain tumour biology about the diagnosis of oligodendroglioma, and the one tumour I haven't talked about, which is oligoastrocytoma, which is supposed to be a hybrid of oligo and astro cells. That's been very controversial for many years, that oligoastrocytoma entity. And one of the great hopes with the new WHO classification and the new molecular markers is that there will be an independent, reproducible test you can apply to that tumour to put it down one diagnostic pathway or the other. I hope that answers your question. Yeah.
All right, well, if you can join me in thanking Kerry and Michael for what was obviously a very informative part of the session.